Good afternoon. Today we are going to talk about mechanisms of pathogenesis. We're going to talk about specific examples of how some viruses cause disease, and we're going to use a lot of what we've learned so far. We're going to be talking about how the immune response collaborates with the virus and causes tissue damage. So pathogenesis, you remember, is a mechanism by which a virus causes disease. So we're going to talk specifically about that. We now have enough understanding to do that. And this quote is very appropriate. We have met the enemy and he is us. And the enemy is the immune response, right? You think it's good, it is good. Without it, we'd be toast, but it causes damage. And that's a problem. Now, to understand how viruses cause disease, we can observe what happens in humans, but we're limited. We can't actually do experiments with people. For most viruses, it's not ethical to do that. And for the few viruses where you can infect people, like rhinoviruses and certain influenza viruses, well, first of all, it's extremely expensive to do that. You can't use a lot of people. Your numbers are limited, and you're limited because you can't really manipulate the host. So we depend on animal models for understanding most of what we know about how viruses cause disease. So I want to talk a little bit about animal models first. And the word model is very important. It is a model. It is not a predictor. It is not a mimic of what happens in humans. And so we have this wonderful saying, mice lie and monkeys exaggerate. Okay, so people think, wow, a mouse is so far from a human, whatever they tell you is going to be wrong. Well, not necessarily. What you find out may be different from what happens in people, but it may be similar, and sometimes what you learn in a mouse allows you to observe what's going on in people and maybe validate it. And other people will say, well, a monkey is close to a human, so that should be better. Well, no, actually. In fact, many things are not the same, even in monkeys versus human. So animals are models for what we learned. They are always models, they are not predictors. Now this slide shows you some of the ways we can use mice to study virus infections. This is by far the most widely used animal model today. A few other animals are used, but mice widely used because they're easy to breed. You can do genetics very readily. And we can infect them and study the disease. And we can infect them with modified viruses and study what happens. Very, very valuable. And some of the ways that we use mice are shown here. Sometimes, as I'll show you in a moment, we can put human virus receptors into mice, because maybe the mice are not susceptible to infection, but once you put a receptor in, they become so. Sometimes people put complete viral genomes in mice, see what happens. Or individual viral genes you could put in to study what the effect is on the animal. Sometimes you can put in clonal T-cell receptors to look at the effect of recognizing one viral epitope. You can deplete immune components. We showed that last time. We depleted B or T cells from an experimental animal. You can do that readily. You can do that genetically or with antibodies. You can either delete cells, entire cells. You can take out B or T cells, or all B, T, and NK cells if you want. There are genetic models for that. You can take out cytokine genes and cytokine receptors. So now you can easily genetically manipulate mice. So you can change things and then infect with your virus and see what happens. You can even overproduce immune mediators if you'd like. So there are lots of things, lots of ways that we can manipulate mice to answer questions about pathogenesis. But what about the virus? What virus do we use? So there are two general areas. First, we take a human virus and we infect the mouse with it. Sometimes that works, but not always. Not all human viruses will infect mice for a variety of reasons. Sometimes we can get around that, as I'll show you in a moment. Sometimes you have to passage the virus in mice. What does that mean? You take a human virus, you infect mice, it replicates a little bit. You take what replicates, you grow it up and infect another mouse, and maybe after 100 of that, passages, mouse-to-mouse passages. You now have a virus that grows well in mice. So obviously that requires patience, right? You can't stop after 5 and say it doesn't work because you have to keep on going. So you can adapt human viruses to mice. On the other hand, sometimes you can use animal viruses. A mouse virus that causes a disease in mice that looks like the human disease. And people do that, and that can be informative as well. So those are the general approaches we take when we use mice to study viral diseases. There are, as I said, there are other animals that are used, but they're not as convenient. For example, some people use ferrets. Anybody have a pet ferret? I've seen people have them on leashes. You know, they're like this long, and they're elongated with a long snout. People use them for certain respiratory viruses because they have a nasal cavity that resembles ours in some ways. Not the snout, but other things in there. Influenza virus. But it's very, very hard to do genetics with ferrets. So mice are much easier in that sense. So a number of years ago in my lab, we made the first transgenic mouse model for a viral disease. This was way back in the early 90s. And my laboratory first identified the poliovirus receptor. One of my first graduate students, Kathy Mendelson, did that. And this turned out to be a cell surface protein, which is an immunoglobulin-like protein. Each of these little domains is similar to the immunoglobulin-like domain that I showed you last time on the Y molecule. We knew that mice are not susceptible to infection. They're permissive. How do we know that? That's a good exam question, but that lecture is way beyond us, so you won't get it back. We know mice are permissive because you can take mouse cells and put RNA into them and the virus will replicate. One round of replication, but they're not susceptible because there's no receptor. So after Kathy had identified the poliovirus receptor gene, we made a transgenic mouse producing the human receptor gene. And when you affect those mice with poliovirus, they get disease. So the only thing between poliovirus replicating in a mouse is the receptor. And you can see this animal has hind limb paralysis. You put the virus in, it gets into the spinal cord and replicates, destroys neurons, causes paralysis. So this was the first transgenic mouse model for a viral disease. And I'm very proud of this because polio vaccines used to be tested in monkeys. Now they're tested in mice. So I have saved thousands of monkeys. Perhaps the greatest accomplishment of my career. I've saved the lives of thousands and thousands of monkeys. Yes, absolutely. They're not chimps, they're old world monkeys, little cynomolgus macaques, but still, we now use this. We don't have to use monkeys. It wasn't my intent at the beginning. My intent was to have a mouse model to study polio, which we didn't have. And when I started my lab, that's what I did. But the side effect was that we used different animals. And of course, we're almost about to eradicate polio, which is really quite remarkable. We'll talk about that later. So this is an example of how you can make a transgenic, but there are many other ways that you can do. There's many other genes you can put in mice to facilitate virus replication. One of the objects of studying pathogenesis is to understand virulence, the capacity of a virus to cause disease in a host. We have avirulent viruses, which when you put it in an animal don't cause disease. We call those avirulent or attenuated. And we have viruses that have high virulence, where you put a small amount in and they kill the animal very quickly, and you have everything in between. All right? We'd like to study this because we want to understand what makes viruses kill hosts so that we can interfere. We quantitate virulence in order to study it. And in any of a number of ways, some of which are listed here. You could simply measure how much virus replicates in the animal by a plaque assay, for example, or some other assay. How long it takes before the animals die, mean time to death, mean time to appearance of signs, measurement of fever, weight loss, measurement of pathological lesions, reduction in blood, CD4 positive lymphocytes. The list can go on and on and on. But the point is, you can do quantitative measurements and get a number to measure virulence of a virus. By the way, while we're on this signs, there are two things that happen when you infect an animal. You get signs and symptoms. And they're different. You should know that they're different. A symptom is what you can feel. So if you have a headache, only you can feel it. No one can see that. Unless you're cranky, I guess they could see it. But it could be from a number of things. Okay, that is a symptom. A sign is what someone can detect. And so they could detect a fever by measuring it, differences in blood enzymes and so forth. But the other things that they cannot measure, those are the symptoms. So if mice have symptoms, we wouldn't know it because they can't tell us. They can't tell us I have a headache after you put that needle in my brain. So you never get that. All you get are signs in animal models, which are things that you can measure. So that's how we quantify virulence. A real key here is that much of the outcome of infection, the virulence, is often caused by the immune response. And you may not know that right now, but you may think that viruses are what are responsible for most of the signs and symptoms of an infection, but in fact they're not. And we'll talk a little bit about that today. It's the immune response. So here are two examples of measuring viral virulence, two different assays. On the left, we have a graph where we're simply looking at survivors. We're infecting mice with two types of poliovirus, type 1 and type 2, and at different days post-infection. This is an easy experiment. You go into the mouse house and you see how many are dead. You take the dead ones and throw them out, and you count them, and that's your assay. Very easy assay. You don't have to do very much. And so you can see here, this type 1 virus doesn't kill anybody. All the mice survive after infection, and the type 2, they're all dead by 11 days after infection. So that's an easy one, survival. On the right is a little more complicated virulence assay. So by this assay on the left, type 1 is obviously not virulent, and type 2 is lethal in these animals. This is a matter of susceptibility. This is not really virulence because the virus is never replicating. So if it doesn't replicate, it's got to be avirulent. So this is kind of an uninteresting example, but it just illustrates using death as a means of assaying. But yes, these mice don't have receptors for the virus, and these mice do, and that's the difference. So that's really susceptibility. And when I talk about genes in us that control virulence, we'll see some susceptibility genes as well. Now on the right, we have an assay where you infect mice here we have 5 different viruses, and these viruses are all neurotropic. They go into the central nervous system, and they cause damage. So what you do is, at a certain time after infection, you take out the brain, you make slices, thin slices, and you look at them under a microscope, and you quantify the amount of damage. This is what pathologists do, right, for a living. They can look and say, aha, those cells are dead because of a virus infection. They can give numbers to the damage. And so that's what this is on the y-axis, the relative neurovirulence score. They put a number to the amount of damage they see, and that can be cell death. It can be cells infiltrating into the region that they're looking at. And here we have 3 different parts of the brain that are looked at for each virus. And you can see the viruses differ in the relative neurovirulence score. Japanese encephalitis virus is neurovirulent in all parts of the brain, and some viruses are not neurovirulent at all in any of them, dengue virus, and some differ depending on the region of the brain that they're causing pathology. So just 2 examples of virulence assays. Another really key concept here is that virulence is a relative property. It is not absolute. So it is influenced by many things: the dose, the route of infection, the species you're using, their age, actually for an animal it should be sex, not gender, because gender is a cultural thing, right? Social cultural thing. So mice, well, I may be insulting mice, but I don't think they have gender. It should be sex and susceptibility to the host. So you cannot compare the virulence of different viruses at all. You can't say Ebola virus is more virulent than influenza virus. Never. Even if Ebola kills 90% of the people, you cannot say it because the route is different, the dose is different, the organs that are infected is different. You just can't say that. And so if you wanted to compare similar viruses, like we have 3 serotypes of poliovirus, if you wanted to compare them, you could, because they're all polioviruses, but you'd have to put them all in the same animal by the same route and the same dose, etc. Otherwise, if you change one thing in the virulence assay, you can't compare them. So that's what I mean by it's a relative property. I can define the virulence of polio in a specific assay and influenza in another assay, and they're independent. You cannot compare them. All right, that's really key. Different routes, different doses, et cetera, all makes it different. The animal model makes it different. And in the end, even if you used mice, you couldn't compare Ebola and polio because, as I said, they're totally different. So here's an example of how one parameter in a virulence determination can make a big difference. And here's the effect of the root. This is lymphocytic choriomeningitis virus, which is a mouse virus that is used very often in virology as a model because it causes disease in mice, and it's well characterized. This is an rhinovirus, by the way. It's an enveloped RNA virus. So here we inoculate intraperitoneally mice, put a needle in the peritoneum, 100,000 PFU, and all the mice live. It's a happy mouse there, right? And if you put 1 PFU intracranially right into the brain, all the mice die. So the route of inoculation makes a big difference. You can't say that LCM is either virulent or not virulent in mice unless you tell me the route of inoculation. That's very important because here you see it makes a big difference. Now you may be wondering why, if you put it IP, it's not lethal. Well, there could be a lot of reasons. We'd have to figure that out. My guess, one guess would be that the virus can't get from the peritoneal cavity to the brain. It can't invade the CNS, but you'd have to do experiments to figure that out, and they'd be pretty straightforward to do. So this is just one example of how the way you do the assay can influence the results. So you can imagine that almost any other parameter, from the simple one to the amount of virus to a major one, which is which animal you're using, would make a difference. So which statement about viral virulence is wrong? It can be influenced by dose, route of infection, species, age, gender, and susceptibility of the host. It can be quantitated by measurement of fever. Ebola virus is more virulent than human papillomavirus. It's the capacity of a virus to cause disease in a host. When comparing virulence, the assays must be the same. Most of you got C, which is the right answer. Ebola virus is more virulent than human papillomavirus. You probably feel that. So the ones of you who answered the other, you probably feel that Ebola virus is more virulent, right? But you can't say that because you cannot just look at what happens in a population and say that one is more virulent than another because they're introduced into people by different ways, different doses, and so forth. All the others are correct. It's influenced by dose, et cetera. It can be quantitated by measuring fever. It's the capacity of virus to cause disease. Assays must always be the same. So you can bet that that'll come up on the next exam. I think that's a really important concept. Now, in virology, what we try and do is understand what controls viral virulence. That's controlled both by virus and host genes. And today I'll tell you a little bit about both. And for both systems, we make mutations. So first, years ago, we were able to make mutations in viruses. Starting from the 50s, when we learned how to do plaque assays, we could mutagenize viruses and pick out mutants and put them in animals. We didn't know what genes the mutations were in, so that made it difficult. But now, of course, we can not only have genome sequences, but we can introduce specific mutations into viral genomes and know exactly what a gene does. We can take a whole gene out of a virus, and if a virus replicates, we can ask, does it have a role in disease? And now we can do this with hosts. We can do that with mice. We can use CRISPR-Cas and make mutations at will in any gene, as I told you earlier. So we make mutations in the virus and the host, or both, and we infect, and we measure virulence by our assay, and we carefully quantify it. So here's an example of an experiment like that. Let's say we have a virus that infects mice. We inject the virus intracerebrally right in the brain. And you'd be amazed, I've done this thousands of times. The mice get up and run around, they're fine. And for us, it would be like putting a vacuum cleaner hose in your head, I think, the equivalent. But mice seem to be fine. Of course, I don't ask them questions afterwards, so I don't know what I've done to them. But this particular virus you put right in the brain. We measure neurovirulence. You could measure paralysis or death, whatever the assay is, doesn't matter. But let's say we determine that this virus is neurovirulent. And at the same time, this virus makes nice big plaques. So that's the wild type, the parental virus that we're using. Then we make a mutant. And we have a mutation in the viral genome that makes the virus replicate poorly. You get smaller plaques and you see the titer is lower. And we put this virus in the brain and it's not neurovirulent, it's reduced neurovirulence. We call it attenuated. But this is not an interesting virus at all because all it's doing is growing poorly. It does so in cell culture and it grows poorly in the mouse brain. It's not interesting because this is just a general growth determinant. It's some gene that's needed for replication. But the next mutant, this is a separate mutation here down at the bottom. This is a mutation where the virus still makes big plaques, it grows well in cell culture, and it's attenuated in mice. For whatever reason, it might not reproduce in mice, but here is a gene specifically needed for virulence in the animal. And that's the kind of gene we're interested in, that will work only in the host and not just in cell culture. Because of course cell cultures are totally different. Cell cultures are cells, no immune system, no other tissue structures and so forth. And in the animals where we want to have our phenotype of attenuation. So when you do this, and people have done this for all viruses over many, many years, what do you find? Well, viral virulence genes fall into these categories. There are some that affect replication, again, specifically in the animal. Some genes are involved with invasiveness, so the ability of the virus to get into a tissue. Remember when we talked earlier on about infection basics, we talked about how viruses get into tissues. Well, mutations in genes can affect that, so getting into the CNS from an intraperitoneal site, that would be invasiveness. Tropism, the ability of a virus to infect a specific tissue, can be affected by viral genes. And then, of course, I've told you in the last few sessions that viruses all encode gene products that modify host defenses. And if you take those out, for most viruses, you definitely see an effect on virulence. Because the virus can no longer antagonize host defenses, it's cleared, so it is less virulent. And those viruses, this is a good example of this, those viruses are fine in cell culture. So if you have a gene that's antagonizing MHC presentation of peptides, it doesn't matter in cell culture because there's no immune response to take advantage of that. There's some genes that enable virus spread in the host, and then there are some genes that are involved in killing the cell. Many viruses do kill cells overtly. They cause cytopathic effects, and that can contribute to virulence, of course, and gene products involved in that, when changed, will alter the virulence. Let's talk about some examples of viral virulence determinants. At the start, I want to tell you that these determinants, they don't have to encode proteins. So they don't have to be genes in the sense that we have a gene that encodes a protein. There are many, of course, but I want to show you an example of one. This is the poliovirus genome, a positive strand RNA about 7 kb long, and we have at the 5' end, remember, there's a little protein linked to the genome, and there's a very highly structured RNA in the first 750 bases. The vaccine strains of poliovirus, there are 3 of them. They're called the Sabin vaccine strains. Those are the ones that we take orally. We'll talk about those in more detail. Those were developed by Albert Sabin in the 1950s. And these were just mutants that he empirically isolated that have less virulence. They were shown to be less virulent in monkeys when he tested them. And when you give them to people, they are less virulent as well, and that they don't cause paralysis. They all have mutations in this 5' non-coding region. So remember, the genome starts translation at about base 740. So this is a non-coding region. It doesn't code for any protein. Yet, in this region, in this stem loop 5, which is expanded on the right here, the 3 vaccine strains each have a single mutation. Here they're shown in red, type 1, type 2, and 3, that reduce the virulence of the virus. It's a non-coding mutation. And so that's just an example of a virulence determinant in a virus. It doesn't need to be encoding a protein. How this works, we don't know to this day. This is actually the effect of that mutation. Here is a assay on the top for lethality. We're measuring the 50% lethal dose. That means the amount of virus that will kill half of the animals. And so that's an endpoint assay using lethality as an endpoint. So we have two viruses which differ only at one of those bases in the 5' non-coding region. This happens to be base 472. The virus with a C is quite lethal. 9,000 PFU, when injected into a mouse intracranially, will kill half of the animals. You change that C to a U, and now you could put over 10 to the 7th PFU intracerebrally, and the mice are fine. They don't get paralyzed. One base change. There's no other change in the genome. It's really remarkable, and this is in a non-coding region. And at the bottom is another kind of assay for virulence. Here we're looking at replication of these two viruses in the mouse brain. So we inoculate the animals, we remove the brain, we grind them up, we do a plaque assay, and we measure PFU per gram of brain. And so you can see that with time after infection, obviously you have to have groups of animals at each time point that you sacrifice and you remove the brain. You can see the virus with a C replicates nicely. It increases till about day 7 or so, whereas the virus with a U really doesn't replicate. It's kind of remaining and then declines slowly at the end. And that's probably why that doesn't cause any paralysis. So that's an example of a non-coding virulence determinant. These kinds of studies have revealed lots of viral genes that encode proteins that modify host defenses. And we've talked about some of these so far. These include genes that antagonize these intrinsic proteins like apoptosis, autophagy, and ApoBec3G, a specific protein. Of course, apoptosis and autophagy are processes, but there are viral genes that antagonize all of this, all these intrinsic defenses, and we find mutations in those viral genes attenuate neurovirulence or virulence because they can't antagonize intrinsic responses. Viruses encode what we call virokines and viroceptors. So mutation of these genes make the virus less virulent. A virokine is a secreted protein that looks like a chemokine or a cytokine. And it will bind the receptor on the host cell, but have no effect. So normally a cytokine or a chemokine will bind a host cell receptor and have some pro-immune effect in the animal. And these viral homologs bind but don't do anything. So they're saturating the receptor so the actual cytokines can't bind to them. And the same way virus genomes can encode what we call viroceptors. These are soluble cytokine receptors. So they float around in the circulation or in tissues and they sop up all the cytokines that are being produced so they cannot get to their target cells and have the intended consequence. And we talked a little bit about what cytokines and chemokines do. So these gene products are antagonizing them. Viruses encode complement binding proteins, which will antagonize complement again. If you mutate them, the virus is less virulent. Modifiers of MHC1 and 2, which we talked about last time. And again, you can take these genes out and the virus grows perfectly well in cell culture because this antagonism is not needed in cell culture. None of these, or few, maybe some of the intrinsic defenses are present, but all the others are not present, so it doesn't matter. So that's an example of a gene where you can remove it. There's no effect on growth in cell culture, yet in an animal there's a big difference. Here's one specific example of a antagonist, and this is in a mouse herpes virus called gamma herpes virus 68. So this is an example of an animal model where the virus is not a human virus. This is a mouse herpes virus, but it causes disease in mice, so we use it as a model to try and understand the human disease. And the gene called M3 encodes a chemokine receptor, a soluble chemokine receptor which is produced in cells, in infected cells. It's secreted and it binds up all the chemokines that are specific for it so they don't have any pro-immune effect. So here we're looking at percent survival. When you infect mice with these viruses, it's lethal. And so here we have percent survival on the y-axis, and we're doing a dose response curve. We're putting more virus in on the x-axis from 0.1 up to 1,000 PFU. And you see the wild-type virus in blue. There's the dose-response curve. You have an S-shaped curve with increasing virus. More mice are dying. And if you want, you could measure the 50% lethality point, which would be slightly less than 10 PFU right there. You could actually calculate that mathematically, but you can see visually it's just slightly less than 10 PFU. Now, if you take out the M3 gene from the virus, you remove it, that's the green virus, delta M3. Now you see you need to get lethality, you need to put a lot more virus in. And if you do the 50% endpoint, right there, it's somewhere, it's maybe 500 or so compared to less than 10. So that's a nice quantification of the virulence of this virus. Removing that one gene, this chemokine antagonist, really reduces the virulence. Now, when you do these kinds of experiments, you are never sure if you're making a change elsewhere in the genome. You know, you can take a big genome like this, 200 kilobases, and pull out a 2 kb gene, but how do you know you're not messing up the rest of the genome? So what you do is you take your mutant and you put the gene back in and see if it restores the wild-type phenotype. If it doesn't, that means there's a mutation elsewhere, and you better redo the whole thing. And so that's this one, delta M3. MR stands for marker rescue. It's an old term which just means we're going to put the gene back in that we took out. And that virus looks just like wild type here. That's the red curve. So we're pretty sure that the effect, the attenuation, by removing this gene is due to that and not something else. So that's an immune antagonist showing how removing it decreases the virulence of the virus. Some viruses actually encode toxic proteins. This is pretty rare in the virus world. Lots of bacteria encode toxins of various sorts, diphtheria toxin, for example, and many others. But viruses don't tend to do that. But here's an example of a virus that does. It's a rotavirus. And these are viruses that cause gastroenteritis. If you've ever had like a 3 or 4 day course of vomiting and diarrhea after you ate in a restaurant, could be this virus. We'll talk more about this later. But that disease, that gastroenteritis, vomiting, diarrhea, is largely caused by a single viral protein called NSP4. So it's a non-structural protein made in infected cells. And here's an intestinal epithelial sheet. If you just take that protein and add it, if you could feed it to an experimental animal, this has been done in animals, that protein alone will cause diarrhea. So that's the source of the diarrhea. And you may say, why would this happen? Well, it helps to spread the infection, right? Having diarrhea, well, you can think about it, right? That helps to spread viruses. And so does vomiting, by the way, especially on an airplane. So that protein alone, what it does, it does two things to cause diarrhea. It inhibits a sodium glucose luminal cotransporter, which is important for maintaining fluid balance. So that's gone, and so you can't reabsorb fluid from the lumen back into the cells, so the fluid goes out as diarrhea. And then this protein increases intracellular calcium, which has a similar effect at causing imbalance of fluids. And so those two activities are responsible for the diarrhea. So that's an example of a toxic viral protein. If you take NSP4 out, no effect on virus replication in cells, but in an animal, it doesn't cause diarrhea. Well, now we're moving on to cellular virulence determinants. And we talked about this before. This is a cellular microRNA. It's miR-122, which is a liver-specific microRNA that's involved in cholesterol metabolism, but it happens to be required for hepatitis C virus replication, which is a liver-specific virus. And there's miR-122 right there on the right. It's the sequence, very short. It's a typical 21 nucleotide microRNA that regulates gene expression. And what it does, it binds to the 5' non-coding region of hepatitis C virus. You can see it binds to two sites, the two purple boxes there at the left end, and at the bottom is a detailed depiction of how the microRNA binds to the viral RNA at those two places. This binding is absolutely needed for the virus to replicate in the liver. And this miR-122 is missing from non-liver tissues, and that's why the virus doesn't grow there. And there's a drug that's been developed called miravirsin. which is complementary to miR-122. And this basically binds to miR-122, hybridizes, and prevents it from binding to the viral genome, so it blocks virus replication. And that's been through clinical trials in humans and shown to be effective in preventing resolving hepatitis C. So in a sense, this is a virulence determinant. It's a permissivity determinant, right, because it's involved in post-receptor interactions. We are now, since we can sequence genomes quite readily, of all kinds, including our genome, we can sequence a human genome for less than $1,000 in a very short time. Now we can take people who seem to get more serious disease when they're infected with a virus than other people and ask, what kind of mutations do they have? Now this is not an easy question to answer, right? Because the human genome is big, even the exome, 1.5% of the 3 billion bases is a lot of bases. But you can get some hints. And so in this example, we take people who get herpes simplex encephalitis. So we'll see in 2 sessions that herpes simplex is a virus that establishes persistent infections. You get a primary infection at a mucosal surface. For type 1, it's typically the mouth. The virus becomes latent in your local ganglia, the trigeminal ganglia, and periodically it's reactivated by stress, sunlight, UV, fever, other kinds of stress. And when it's reactivated, you get a fever sore again, so the virus travels from the ganglia back to the mucosal surface. But sometimes, in some people, the virus gets into the CNS and causes encephalitis, which is an infection of the brain, and this can be serious and can have long-term sequelae, destroys neurons, and you have issues for the rest of your life. So it's a rather rare infection, about 1 in 250,000 cases of simplex every year. So this is a rare individual that gets this, has a high mortality if you don't treat it, but we can treat it, but again, you can have long-term sequelae anyway. And we see 2 peaks of incidence of herpes encephalitis at early age, which is the primary infection. You get the virus from your parents, for example, and sometimes that goes right to the brain and causes encephalitis. Or people greater than 50 years of age, as the virus reactivates from latency, instead of just causing a fever sore, it goes into the brain. So the question is, what's different about those people? So a number of groups have sequenced the people's genome. There's one group here at Rockefeller that does that, and a few others. And if you sequence enough people and look at family pedigrees, you can figure out what's going on. So in this case, there are genes involved in the toll-like receptor 3 pathway. Now, this whole process of figuring out what mutations are responsible for increased susceptibility, in this case to getting herpes simplex encephalitis, we do genome-wide association studies, which means we sequence people's genomes and we say what mutations go along with encephalitis. It's not easy because a lot of mutations are going to appear to go with it, but if you sequence enough people and if you look in certain pathways, you can do it. And what you're looking for are SNPs, single nucleotide polymorphisms. We don't like to think of the population of people of having mutations. It's not, it's just variation from person to person, so we call those polymorphisms. So we do genome-wide association studies, sequence genomes, see what single nucleotide polymorphisms go with encephalitis. And mutations have been found in the genes encoding toll-like receptor 3, which is a toll-like receptor that detects viral nucleic acid, and other proteins that are involved in that pathway, UNC93B, TRIF, and TRAF. And you can see on the bottom here where these are working. TLR3 is a endosomal TLR. It detects viral nucleic acids in the endosome. It then signals through TRIF and TRAF3, which isn't shown here. So mutations in either TLR3 gene, TRIF, or TRAF affect the ability of these individuals to respond to a herpes infection. So you get higher titer viruses, it gets in the CNS, causes encephalitis. UNC93 is a protein that's needed for trafficking of TLR3 in the cell. So again, if you have a mutation in UNC93B, prevents TLR3 getting to the endosome where it needs to be. So here, it's a beautiful study where you can actually say that these mutations make these people more susceptible, because what you can do is put the mutations in say, TLR3 in cell culture and show that herpes viruses replicate better. Often we take fibroblasts or epithelial cells from these individuals, put them in culture, show that the virus grows well, and then add back the wild type gene and show that it reduces viral replication. You could do an amazing number of things to pinpoint what's going on. So that's one example. I want to give you one more, a more recent one, where, and this is a case of a young child who from birth had recurrent respiratory virus infections. And so here's the timeline of this child here from birth to 5 years of age. And each triangle or circle is some kind of infection. So this is not normal to have so many infections within the first few years of life. I mean, you do get 1 or 2 a year normally, but this is completely wrong to get this. And you can see at the top here, rhinovirus. So they do respiratory fluid sampling, and they can say what viruses are in this person. Lots of rhinovirus infections, coronaviruses, influenza virus, respiratory syncytial, adenovirus, and even some bacterial infections. And so they sequenced the genome, the exome of this child and the child's parents and siblings. And they found a mutation. So here's a trace of the DNA sequence, which shows a difference in the child compared to the parents and siblings. So that's the child right there, the proband. And this is MDA5. It's a cytosolic helicase that recognizes nucleic acids as part of the innate immune pathway. And single amino acid here in this part of the protein causing a single base change causing a single amino acid change. And if you look at the pedigree, here's the child, homozygous for this mutation. Parents are both heterozygous. They have no phenotype because one copy of this gene is enough for it to function as an innate sensor of infection. And one of her siblings was also heterozygous, right? So this is what you can do. Of course, you need the family's cooperation. You have to follow this child for a long time. They have to be cooperative. Some people are willing to do that. But in this case, the conclusion is that MDA5 is really important for sensing rhinovirus infections. It seems to be less important for the others. Even though she had other viruses, it's not clear if other changes weren't responsible for those as well. But MDA5 is important. If you can't sense the infection, the virus grows to high titer, and you get serious and recurrent infections. So there's the reference if you want to check that out. So people are doing this all the time. It's quite interesting. Now here's an example of another host gene, which I say determines susceptibility because it's at the level of receptor. The outcome is that the virus is not virulent in certain people, but it's because the virus cannot get in. And this is in the protein called CCR5, which is a co-receptor for HIV. Now if you remember way back to the early parts of this course, we talked about how HIV, via its surface glycoprotein, can bind to two receptors on the cell surface, CD4 and a chemokine receptor, CCR5 or CXCR4. There is a fraction of the human population that lack the gene for CCR5. It's called delta 32. It's actually an out-of-frame deletion mutation which blocks synthesis of the protein. 4 to 16% of the world's population, these are mostly people of European descent. They don't make CCR5, so apparently you don't need it. Why they're around, we don't know. We don't know what was the selection pressure that led to their emergence. People think it was a pathogen many, many years ago that selected for people who lack this gene, but we don't know what the pressure was. However, these individuals are resistant to HIV infection because the virus can't get in. They're not susceptible to infection. So that has led to the idea of trying to make people resistant or cured of HIV by deleting CCR5. And now we can do that with CRISPR, but of course we can't do that with people yet. So you can't do it in the germline anyway, but you can take out T cells and knock out the CCR5 gene and put them back in. We'll talk about that later. But what was done a number of years ago with a German patient, it's called the German AIDS patient. This guy had leukemia and AIDS, so they gave him a bone marrow transplant. They irradiate you to destroy all of your bone marrow, so all the precursors of your lymphocytes are gone. And then they give you a transplant from someone else, called an allogeneic transplant. Now, what the doctor did, and that's to cure his leukemia, but the doctor said, well, while I'm at it, let me get a donor that is lacking the CCR5 gene. So he found one, and they gave this patient compatible cells that lack CCR5 gene. He was the only person ever cured of AIDS. Since then, he has no more virus in him. Actually, he needed 2 transplants. The first one didn't work. But after the second, he had no more virus in him because all of his T cells are now coming from his new bone marrow, which doesn't have CCR5. So for many years, he was the only patient. People tried it and never worked with anyone else. And then just this last month, it was reported there's a second patient called the London patient now, who has also gone through the same thing, 1 bone marrow transplant, and it's worked in him as well. And I hear that there are some others that are incubating as well, but they want to wait a while to say that they're virus-free. So that's an example of how you can attenuate. You're not really attenuating the virus, you're removing the receptor from the host, but in essence, you're preventing disease. There are other determinants of virulence in the host. I want to tell you about, we really do not understand how they work. One of them is age. In general, very young people and very old people are more susceptible to viral disease than people in the middle in terms of age. And we think it's because in young people, the immune response is not yet mature, so it can't handle the infection. In old people, it's diminishing. The immune responses are diminishing in effectiveness. In addition, things like our alveoli become less elastic. We're less able to expel viruses. Weaker respiratory muscles, diminished cough reflexes, and reduced rate of production of new immune cells. I learned recently that after over 55 years of age, most of your bones don't have any bone marrow in them anymore. I didn't know that. So you cannot make many new lymphocytes. You have some memory cells to previous infections, but a new infection is going to be difficult. When I heard that, I felt like I was a shell of a human. I have no bone marrow. Well, a little bone marrow, but not very much. And so that's another reason why old people are more susceptible. So here's a graph that illustrates that nicely. This is the death rate from influenza in the US from 1911 to 1915. And it's stratified by age group. And you can see the very young had a high mortality rate, in between not so much, and then very old had a high mortality rate as well. So it perfectly reflects the very young and the very old being highly susceptible to infection. So that's age. That's one determinant. How about sex/gender, which we can talk about for people? Sex is a biological property. Gender is a social construct which influences your behavior and hence influences how you may be infected. In general, men are slightly more susceptible to viral infections than females, women, but not always. There are exceptions. We don't know why, but if you just look at all viral infections in toto, that's the general trend that you see. It's been pretty much not studied until the last few years, so people are just starting to understand why. Here's an exception. Pregnancy makes women immunosuppressed, so hepatitis A, Hep B, and influenza are all more lethal in pregnant women compared to non-pregnant women. But as I said, we don't really understand the specific mechanism. So that's sex and gender. Malnutrition is a big one. It increases the susceptibility to infection. It compromises the immune response. It compromises physical barriers. An excellent example is measles. In developing countries, measles is 300 times more lethal than in the US and Europe because the kids are malnourished, and that makes them more susceptible to infection. They would love to have a measles vaccine. Some other determinants. Cigarette smoking increases your susceptibility to respiratory infections. Air pollution, if you live in a city with a lot of air pollution. New York City has been cleaned up in the last 20 years, it's pretty good, but there are other cities that are not, a lot more respiratory disease. And of course, stress as well, and for reasons we don't really understand. And I love this sign, right? Stress spelled back. I don't know what it has to do with viruses, but it's fun anyway. And lastly, something we have to remember is our microbiome, and in fact, our virome as well. but this can have an influence. So here's an experiment in mice. You have poliovirus receptor transgenic mice, and when you infect them, the virus replicates. So here we have untreated mice viral replication. After oral inoculation, the virus replicates well. So we're not looking at paralysis, just virus replication. If you give these mice antibiotics and deplete their gut microbiome, you can see on the left, so we're taking their feces and measuring colony-forming units of bacteria. And untreated mice have lots of bacteria in their feces. Antibiotic-treated mice, ABX, you see the bacteria counts go way down. If you take those antibiotic-treated mice and feed them poliovirus, the virus does not replicate. Those are the squares here. The virus can't replicate without a microbiome. So this is part of susceptibility or permissivity determinant. We're not sure exactly what's going on here. There's some hint that components of bacteria are binding poliovirus and stabilizing it in the gut. And makes sense to me because the virus has evolved to infect the gut along with all those inhabitants, microbial inhabitants. So we can't ignore this, the microbiome, and the virome as well, I'm sure makes a difference in susceptibility to infection. All right, now let's talk a little bit about what actually causes disease in a virus infection. We've talked about how viruses kill cells. We talked very early on about cytopathic effects, the lysis of cells in cell culture, the formation of syncytia, the fusion of cells to make one giant cell. That is cell killing. And this is done by cytolytic viruses. They can have these effects. They can cause apoptosis, necrosis, pyroptosis, different kinds of programmed cell death. And that contributes to the pathology. These viruses encode a variety of proteins that do this. They encode viroporins, which form channels in the cell and have the contents leak out so that they die. They inhibit a variety of synthetic processes in the cell, which damages the cell, and syncytium formation as well. So that is what cytopathic viruses can do when they get in you. They can destroy cells, and that's part of the pathology. But it turns out that the immune response makes a bigger contribution than this. But first, we have to answer a question. All right, which statement about determinants of viral virulence is incorrect? Virulence genes can encode viral proteins. Virulence genes can encode cellular proteins. They are the same in all viruses. They can be found in untranslated regions. They may encode immune modulators. Okay, most of you got C, which is the right answer. They're the same in all viruses is not correct. Virulence determinants are not the same. Some of you answered B or E. Virulence genes can encode cellular proteins, and they may encode immune modulators for sure. I've told you that in the past few minutes. So let's, for the rest of the time, focus on immunopathology. That is what the immune response does to contribute to pathogenesis. I call it too much of a good thing. So the clinical signs and symptoms of a viral infection, these include fever, tissue damage, aches, pains, nausea. For most infections are mainly a consequence of the immune response. There's a contribution of cell killing, but as you'll see, some viruses don't kill cells. So all the symptoms are caused by the immune response. So on cytopathic viruses, the disease is always a consequence of the immune response. And you can remember the cytokines that we talked about earlier that cause the 4 signs of inflammation: fever, redness, swelling, pain. Those are caused by the immune response, not by viruses. So we know for a number of viruses exactly what part of the immune response causes immunopathology, and here's a listing of them. We know for some viruses it's CD8 positive T cell mediated. For some it's CD4 T cell mediated, and Th1, we can divide it even further. For some it's Th2 CD4 positive, and for some it's antibodies. So I want to show you a couple of these and how they work. So again, this is immunopathology. You have a virus infection, the immune response is causing the disease. So here's an example with lymphocytic choriomeningitis virus, a virus that is a mouse virus that we use to study viral pathogenesis. This disease is mediated by CD8 positive cytotoxic T lymphocytes. You take an adult mouse, you infect it with LCMV. Here we're putting IC. within 8 days the mouse is dead from a lethal infection. If at the time of inoculation you immune suppress the cell, you can give it a global immune suppressant so that every part of the immune response is suppressed. These mice survive and they have a persistent infection for as long as they live, but they're fine. So apparently the virus is not killing them. If you then give them back different parts of the immune system, when you put back CD8 positive CTLs, which are virus specific, remember they have to recognize a viral peptide, then the mice die. So the CTLs are actually killing the mice in this particular example. And I have a little more information on the kinds of lesions that are associated with these lymphocytes. This is another kind of, this is the same virus, which is now infecting the heart. And so that's what's shown on the bottom. Top left, we're looking at survival after infection with wild-type mice and with mice lacking the gene for perforin. Perforin is one of the pores that the CD8 CTLs inserts into the target cell to kill it. So we can take the perforin gene out of the mouse. Look, these mice survive LCMV infection. So before, remember, I showed you if you infect mice with LCMV, they all die unless you immunosuppress them. Well, you can narrow that down to perforin, which is the protein that CTLs use to kill cells. So the perforin null mice survive. The same experiment, you can measure liver enzymes to look at liver damage. So wild-type mice, liver enzymes leak into the circulation, the sign of liver damage. The perforin knockout mice, that doesn't happen. And on the bottom, you can look at the heart tissue, which also gets infected. On the left, you see this blue staining indicates a certain kind of pathology in the heart. These are wild-type mice. The perforin knockout mice don't have that heart damage. So, beautiful example of how a specific component of CD8 CTLs is responsible for immunopathology. CD4 positive cells also are involved in immunopathology. Remember, these are helper cells that can make Th1 or Th2 cytokines. So they make a lot of cytokines, which can recruit what we call non-specific effector cells. And these include neutrophils and mononuclear cells that can cause cell damage in their own. So the CD4 Th cells are making cytokines. They're recruiting cells that cause damage in the area. And these cells release proteases, reactive radicals, cytokines in themselves, the neutrophils, the mononuclear cells, and they cause cell damage. So let me show an example of that. This is herpes stromal keratitis. It is blindness associated with herpes simplex infection. This can happen. If you have a fever sore, you shouldn't touch it and put your finger in your eye. You might get herpes stromal keratitis. It's one of the most common causes of blindness in developed countries. And it is pretty much all immunopathological, caused by CD4 positive Th1 cells. What happens is you get repeated infections of the eye, and eventually the cornea becomes opaque and you can't see. So that's what we have at the bottom here. This is a person whose eye has blindness as a consequence of herpes simplex infection. So how does this happen? So let's take a cross-section of the cornea, and you can see that on the left here. So on the top is the corneal epithelium, and the bottom is the cornea itself. The virus replicates only in the epithelial cells, but the damage occurs underneath in the cornea proper. The virus doesn't replicate there, but replication attracts CD4 positive Th1 cells. Those cells are in the underlying stroma. You can see them staining darkly there. And they secrete cytokines that recruit other cells in that damage the stroma. It's the same on the right here. The epithelium is at the top, and the inflamed stroma on the bottom. There's no virus in this corneal stroma, but there are cytokines in Th1 CD4 cells, and that's causing the damage. So nice example of immunopathology. Another one, West Nile virus. In a certain percentage of people who get West Nile infection, remember, this is a mosquito-transmitted virus that in a certain fraction of people will get into the brain and cause encephalitis. It turns out that, at least in mice, getting in the brain is a consequence of cytokines. And in particular, TNF alpha is one of the cytokines produced after the virus is sensed. Cytokines are produced. One is TNF alpha. It compromises the blood-brain barrier, lets viruses in. The point is not to let viruses in, of course. It's to let cells be able to traffic, but the virus is going to get in. So this was determined because mice lacking TLR3, which is a major sensor of West Nile virus, they're more resistant to lethal brain infection than wild-type mice because the virus cannot get into the brain, because if you knock out TLR3, they're not going to make TNF alpha. And that's an experiment to show that. Here these are mouse brains, and they're all infected with West Nile virus. So the mice were infected, and then at a certain time after infection, the brains are removed. And along with the virus, they put a dye in, and if the brain becomes permeable, you can see the blue dye in the brain. So it's a nice visual way of seeing that. So here we have wild-type mice, day 0. By day 3, brains are blue. That means TNF alpha has been produced and the blue dye is getting in the brain. It's a nice visual way to see that. Here's TLR3 knockout mice. They don't have as much permeability on day 3. As you can see, it takes longer to get a little bit of permeability. So TLR3 knockout, they don't sense the virus, they don't make TNF alpha, it doesn't permeabilize the brain. So the virulence is affected by this immune component, TLR3. As you know, we've talked before about how many virus infections cause poxes and rashes, smallpox, measles, etc. This is a child with measles, rash all over the body. These rashes are typically immune responses to infection. It's not virus alone replicating in cells, it's an immune response. What happens is certain immune cells, Th1 cells, macrophages, are homing in on the foci in the skin because virus is replicating there. But then they produce cytokines, and these cytokines increase capillary permeability. T cells come in, so that causes redness and swelling, makes the rash. So the rash is an immune response to infection. Here's an example of how antibody can cause immunopathology. So far we've talked about cellular immune responses being evolved. This is antibody. This is dengue fever, also called breakbone fever. This is a flavivirus transmitted by Aedes aegypti. It's endemic in many parts of the world where the mosquito is prevalent. Many infections a year, actually second only to malaria among insect-borne diseases. Now this infection is quite prevalent where the mosquito is prevalent. In the top map, the red areas are dengue areas which coincide with Aedes aegypti. So we don't have much dengue. We have mostly imported dengue in the US, maybe a few cases in Texas and Florida, locally transmitted. Now before 1981, we used to use DDT. And we got rid of a lot of mosquitoes by doing that in many parts of the world. This is dengue in South America prior to 1981. And of course, we realized that DDT is not good, so we stopped using it. And by 2003, the mosquitoes were back. That's the red coloring. And they're spread by tires. Your tire wears out on your car, you sell it to someone for $1, and they put it on a container ship and they send it somewhere where it's recycled. But it always has water in it. You can't get it out, right? People don't cover the tires, so they get full of water, and mosquitoes breed in the water. So they start in Florida, and they end up somewhere else in the other side of the world, and they brought the mosquitoes with them, or vice versa. So tire movement is responsible for spreading mosquitoes globally and for bringing dengue back to many areas. But anyway, this is a virus that when it infects you, you get a primary infection. It's either asymptomatic or a febrile illness with severe headache, back, and limb pain. Particularly your bones hurt. They're very, very much like an arthritic disease. Normally you recover. Rarely you can get hemorrhagic fever where your capillaries are bleeding, 1 in 14,000. And that bleeding can lead to shock syndrome. You lose fluids. Now, there are 4 serotypes of dengue virus. When you get infected with one, you make antibodies to that serotype, but they don't protect you against the other 3. And that's where the problem arises. When let's say you get dengue and you get joint aches and bone aches, but you're going to recover and then you get another dengue infection. So now you had your primary infection, you made antibodies against that, and you get a secondary infection. Let's say you're first infected with dengue 1, and now you get infected with dengue 2. The antibodies that you recall by memory against dengue 1 will not neutralize dengue 2, but they will bind dengue 2, and the FC portion will bind to FC receptors on normally not susceptible cells, and like a macrophage, and these viruses will infect them. You get more virus load as a consequence, and the idea is that macrophages are going to release lots of cytokines and chemokines as a consequence, and that gives you the increased frequency of hemorrhagic fever and shock syndrome. Remember, it was 1 in 14,000 in primary infections. Now it's 1 in 90 and 1 in 50. So this is why we care about dengue, because many people have already been infected, and if they get infected again, the mortality rate is quite high, and it's why there is a dengue vaccine and others are in development. Finally, I want to talk about immunosuppression, which is another consequence of virus infection. We don't think of it that way. If you think immunosuppression, you think, well, someone's getting an organ transplant, and you're immunosuppressing them so they don't reject it. Well, it turns out that many viruses are immunosuppressive, and that's presumably because they replicate in immune cells, or it helps them to spread. So you have a global reduction of the immune response. That's what I mean by immunosuppression. It can happen by replicating in one or more cells of the immune system. We'll see HIV-1 later, replicating in CD4 positive T cells, completely eliminates their function. It perturbs the production of cytokines, and some viruses encode virokines and viroceptors, as I say, which can also immunosuppress. So here's an example of that during measles infection. So again, here's our child with measles. And this study was done with children who had measles, and they were studied different times after the appearance of rash. So what they did was to ask, are these children immunosuppressed to a different immune response? And the immune response was the tuberculin test for tuberculosis, where they inject intradermally a small amount of antigen. And if you have previously been infected with TB, you will get an immune response there which causes a swelling that can be measured. So if you're positive, it means you've had or you still have active TB. If you're negative, that you weren't infected. So that's the tuberculin test. So what they do is they take these kids at different times after the rash appears, and they give them a tuberculin test. And so here's the base result. This is induration, the size of the swelling in millimeters. So there is without measles. And then at the time of the rash, if you give kids a TB test, there's hardly any and these kids are TB positive, there's hardly any rash, any swelling of the TB test. And then as you go further out after the rash, you can see that reactivity returns as the original infection subsides. So this shows you that measles infection, as measured by presence of rash, suppresses the tuberculin response. Measles also replicates in APCs, antigen-presenting cells. So normally an APC would recognize a viral antigen, right, and respond by producing cytokines that can influence CD4s becoming Th1 or Th2. And so, as we said last time, for many virus infections, you want a Th1 or an antibody or a CTL response, which will kill infected cells. Measles actually infects these APCs and inhibits the production of cytokines that would favor a Th1 response. It shifts these cells to a Th2 response. So remember, the APC is presenting the antigen to the CD4, and the interaction would determine whether the CD4 makes Th1 or 2 cytokines. Measles is infecting the APC on top of that, blocks the production of Th1 favoring cytokines, it drives it to Th2, and this is not good for eliminating the infection. So that's why the measles is immunosuppressive, and it persists as well. And these are some viruses that can immunosuppress. Measles, we've talked about replicating in APCs. It also can replicate in thymic cells. And then you get enhanced other infections. Kids with measles often get other infections as a consequence. Rubella virus replicates in lymphoid cells and causes persistence of its own infection. And HIV, of course, replicates in CD4 T cells and monocytes, completely suppressing immune responses, and so eventually you die of cancers or other opportunistic infections. All right, so next time what we're going to do is to start looking into the two kinds of patterns of infection. We have viruses that replicate quickly and are resolved. We call those acute infections, and we'll talk about that next time. And afterwards, we'll talk about viruses that stay with you for your lifetime.